What's good, everybody? Welcome to a video on the basics of biochemistry. Now, I'm going to make this as accessible to everyone regardless of your education as possible. So biochemistry is just the chemistry of biology. The first concept that's important to note about biochemistry is the idea of hydrophobic versus hydrophilic. Now, hydrophobic just refers to something that is water hating, and hydrophilic refers to something that is water loving. And 60% of the body is water, which means that all the reactions that are taking place in the body are using water as a solvent, or the thing to kind of immerse everything in. That's why the hydrophobic or hydrophilic nature of a compound is very important. Furthermore, the body takes advantage of these two properties to compartmentalize things within itself. This is because of the concept in biochemistry in which like things dissolve other like things. Hydrophobic or water-hating molecules are able to interact with other hydrophobic molecules, whereas they will repel hydrophilic molecules. You can think of this concept as if you have a glass of water and you pour any kind of oil into that water, the oil will sit on top of the water and it won't mix with the water at all. Even if you shake up that glass of water, eventually those two, the water and the oil, will separate out. These same kind of interactions occur on a much smaller level within your body. For example, cells have a plasma membrane which is made up of fat, so hydrophobic material. And any other compound that is hydrophobic or water-hating is able to easily pass through the cell membrane and react with all the contents of a cell. However, anything that is water-loving or hydrophilic needs to utilize receptors in order to get through the plasma membrane and into the cell. The next properties that are important to know about molecules within the body are the difference between nonpolar, polar, and ionic molecules. Now, nonpolar molecules are simply ones in which atoms that are bound together share their electrons equally between the two atoms. Generally, nonpolar molecules are also hydrophobic molecules, so they have equal charge distributed throughout the molecule. Polar molecules, in contrast, have atoms present within the molecule that unequally share electrons. So one atom will hold electrons significantly harder than another atom. And this causes kind of a slight charge on either side of the molecule, which helps it to interact with other polar molecules, almost like a magnet having a north and a south pole. It has this positive and negative charge where the negative charge of one polar molecule will interact with the positive charge of another. Generally, polar molecules are also hydrophilic. The last kind of bond or sharing of electrons is an ionic bond. And actually, this is where two different atoms just completely exchange electrons. So one atom is giving up its electron and the other atom is receiving the electron, causing an even stronger charged particle. So the atom that gives up its electron will be fully positively charged, and the atom that receives the electron will be negatively charged. And just like in polar molecules, these positively charged particles will generally want to interact with negatively charged particles and vice versa. Now, understanding the different properties that molecules can have will help us to understand the four groups of common macromolecules within the body, which are proteins, carbohydrates, lipids, and nucleic acids. Now, proteins are, in my opinion, the most important macromolecule of the body because it makes up the foundational structure of most cells, carries out most of the functions in the body, and regulates the body tissues and organs. Proteins are made up of a string of amino acids. You can see the basic structure of an amino acid here. It is a carbon molecule with four groups that surround it. There's usually a hydrogen, a carboxyl group, an amino group, and an R group. The R group is the group that varies from the different amino acids. And this is what gives each amino acid a different property, which allows it to bind uniquely to other amino acids or other molecules. So you can see on this next diagram that a protein is a string of amino acids, and then based on the hydrogen bonding of those amino acids, either an alpha helix or a beta sheet 3D shape is created, and then from there, alpha helix and beta sheets interact with one another based on the R group properties of their amino acids, and then once they've created this shape, they can interact with other proteins to create larger structures. Properties of the R groups ultimately creates the shape of a protein, and the shape of the protein determines its functions as to what it can bind to or what it can't bind to. Furthermore, tying that in, I'm going to show a diagram here of the R groups of the amino acids split into the various properties that we were talking about before, that being nonpolar, polar, and ionically charged. So you can make an inference as to what amino acids bind to what amino acids based on these properties. So nonpolar amino acids would probably bond with other nonpolar amino acids, polar amino acids would bond with other polar amino acids, and positively charged amino acids would bind to negatively charged amino acids. This also then, on a larger scale, other molecules within the body, whether it be drugs or just other molecules floating around in the blood, are able to bind to each of these amino acids based on their own properties. So if it's a drug that is a nonpolar or hydrophobic drug, it will most likely bind to the protein at a spot with nonpolar amino acids. If it's a negatively charged molecule or polar charged molecule, they'll bind appropriately. Moving on to nucleic acids, these are, in my opinion, the second most important macromolecules, and that is because they control the proteins that are expressed within each cell. Now, the structure of nucleic acids don't matter as much, but they do form DNA and mRNA. Now, DNA and mRNA are just molecules that are used as templates that the ribosome can read to tell the ribosome what protein to create. So DNA is housed within the nucleus of the cell, and it's converted into mRNA in a process called transcription, and then from there the mRNA is read by a structure called the ribosome, and this ribosome will create the protein that the mRNA has instructed it to. This process is called translation, and translation is important because every cell is specialized to perform a distinct function, and generally the variants in the proteins that are expressed within that cell will contribute to its unique functions. So a muscle cell will express different proteins than a lung cell or a liver cell or an intestinal cell. Furthermore, based on the conditions present within the body, such as stress or nutrients or oxygen, the DNA can communicate to produce certain proteins to rebuild the distinct proteins within each specialized cell. Moving on to carbohydrates. Carbohydrates are molecules that are primarily used as energy, but are also sometimes added on to proteins to act as different regulatory or signaling molecules. But mostly they're just used as energy to produce ATP. And the properties of these molecules are polar and hydrophilic. The last macromolecule we'll go over is fats, which are mostly nonpolar hydrophobic molecules, and these are also used as energy similar to carbohydrates, but they can also be signaling molecules, hormones, form structures as well. They have a variety of functions. Putting all that together, we can understand that based on the properties of various molecules and macromolecules within the body, they will bind, interact, share electrons, create new compounds, and perform all the functions needed to run the body. Oftentimes, these reactions require energy in order to go all the way to completion. And the main currency of energy within the body is ATP. And ATP creates energy by going through hydrolysis, cleaving off one of its phosphates, and the energy used from cleaving off the phosphate from it goes towards completing reactions. Last, in addition to the energy provided from ATP or the splitting of other molecules, proteins called enzymes are used to speed up reactions. So enzymes will bring in two different molecules as substrates. They will lower the amount of energy required to complete whatever reaction is occurring, and then they'll spit the new molecules out. So I hope you guys enjoyed this video. I tried to keep it both high level, informative, but also applicable and understandable to any level of education. If you do have any questions or comments, please leave them in the comments below. Like, subscribe, and have a good rest of your day.